Look at a single cell from your body. Inside it, you'll see something remarkable. The nucleus, the control center, your DNA. And surrounding it, hundreds of smaller structures. Mitochondria. These are not originally you. They're the descendants of bacteria. Ancient invaders that took up residence inside your ancestors' cells 2 billion years ago. You are not a single organism. You are a community. A society of 2 ancient life forms that merged so long ago, you can't exist without each other. Your nucleus, descended from Archaea, ancient microbes. Your mitochondria, descended from bacteria, different domain of life entirely. Every cell in your body contains both. Every breath you take requires both. Every thought you think needs both. And it's not just you. Every complex organism on Earth is the same. Every animal, every plant, every fungus, all eukaryotes, all two-organism hybrids. But here's the mystery. How did this happen? Because the gulf between simple bacteria and complex eukaryotes is staggering. A bacterial cell is elegant in its simplicity. Take E. coli, a typical bacterium. One circular chromosome, floating free in the cytoplasm. No nucleus, no compartments, no membrane-bound structures. Size, about 2 micrometers long. Genes, around 4,000. Internal organization, essentially none. DNA transcription and protein synthesis happen in the same space. No separation. No regulation beyond the immediate biochemical environment. It's like living in a studio apartment. Everything happens in one room. Now look at a human cell. It's not just bigger, it's a different universe. The nucleus. DNA wrapped around histone proteins, organized into chromosomes, enclosed in a double membrane. The endoplasmic reticulum, a vast network of membranes where proteins are synthesized and modified. The Golgi apparatus, protein packaging and shipping department. Lysosomes, recycling centers with digestive enzymes. Peroxisomes, detoxification units. The cytoskeleton, protein scaffolding giving the cell shape, enabling movement, transporting cargo. Mitochondria, the power plants, generating ATP through aerobic respiration. A typical eukaryotic cell is 10,000 times larger by volume than a bacterium. 10,000 times. A human cell? 20,000 plus genes. Complex regulation, introns and exons, alternative splicing. The difference isn't incremental, it's categorical. And here's the problem that's haunted biology for decades. There are no intermediates, no halfway organisms, no simple cells with just a nucleus, no bacteria with just a few mitochondria. You're either prokaryotic, simple, small, no nucleus, or you're eukaryotic, complex, large, compartmentalized. There's nothing in between. Why? For decades, the lack of intermediates was a puzzle. Then, in 2010, biochemists Nick Lane and Bill Martin proposed a radical answer. It's not that intermediates didn't evolve, it's that they couldn't evolve. The reason? Energy. All life runs on ATP, adenosine triphosphate, the universal energy currency. Bacteria generate ATP across their plasma membrane. They pump protons out, create a gradient, and ATP synthase harnesses that gradient to make ATP. It's elegant, efficient. but it's limited by surface area. As a cell grows, its volume increases faster than its surface area. This is geometry, square cube law. Double the radius, surface area increases by 4 times, radius squared. Volume increases by 8 times, radius cubed. A bacterial cell that tried to grow 10 times larger would have 100 times more volume, more cytoplasm demanding energy, but only 100 times more surface area for ATP production. The energy per unit volume drops. You can't support the larger cell. A bacterium has about 200,000 ATP synthases per cell, supporting 4,000 genes. That's 50 ATP synthases per gene. A eukaryotic cell with 20,000 genes would need 1 million ATP synthases. But if you just scale up a bacterium, you only get surface area for maybe 200,000. You're 5 times short on energy. The cell starves. Prokaryotes hit an energetic ceiling. They cannot scale up. They cannot complexify, not without solving the energy problem first. Life appeared on Earth about 3.5 billion years ago. For the first 1.5 billion years, it was all bacteria and archaea. Simple, small, prokaryotic. Around 2 billion years ago, something changed. Cyanobacteria began producing oxygen through photosynthesis. Oxygen levels rose. The atmosphere transformed. For anaerobic life, oxygen was poison. Mass extinction. But for organisms that could use oxygen, aerobic respiration. 20 times more energy per glucose molecule than fermentation. Fermentation, 2 ATP per glucose. Aerobic respiration, 38 ATP per glucose. Somewhere in the oceans, 2 organisms were about to collide, an archaeon and a bacterium. The exact date is debated, but most estimates between 2.5 and 2 billion years ago. Recent molecular clock analysis suggests eukaryotes emerged between 3.0 and 2.25 billion years ago, with the archaeal host already possessing complex features before mitochondrial endosymbiosis. One merger, one evolutionary event, and every complex organism that exists today descends from it. Why only once? If the merger was so successful, why didn't it happen multiple times? Because endosymbiosis between prokaryotes is vanishingly rare. We know hundreds of endosymbiotic relationships. Eukaryotes hosting bacteria, eukaryotes hosting other eukaryotes. But prokaryote hosting prokaryote? Virtually unknown. There's one tentative case in cyanobacteria. That's it. Prokaryotes don't have phagocytosis. They can't engulf other cells. Their rigid cell walls prevent it. So how did an archaeon swallow a bacterium? The answer suggests the host wasn't a typical archaeon. It was already different. 2015. Scientists analyzing sediment DNA from the Arctic Ocean make a discovery. A new lineage of Archaea. They call them Asgard Archaea, named after the mythological realm of Norse gods. Because these Archaea have genes that shouldn't exist in prokaryotes. Typical Archaea, simple metabolism genes. Asgard Archaea, genes for actin-like cytoskeleton proteins, membrane remodeling proteins, vesicle trafficking systems, ESPS, eukaryotic signature proteins. The molecular machinery needed for complex cell structure. 2023. After years of effort, researchers finally culture and image a living Asgard archaeon, Archaeum syntrophicum, long, tentacle-like protrusions extending from the cell, not a rigid sphere, a dynamic, shape-shifting cell. These protrusions could wrap around other cells, envelop them. This is how an archaeon could engulf a bacterium, not phagocytosis in the eukaryotic sense, but entrapment, enclosure. Asgard Archaea already had complex features before mitochondria. The host wasn't simple. It was on its way to complexity.
But why would an archaeon enclose a bacterium? What was the initial interaction? 1998. Bill Martin and Miklos Müller proposed the hydrogen hypothesis. Imagine an anaerobic environment, low oxygen, high sulfur, like deep-sea vents. An archaeon that produces methane, a methanogen. Its metabolism requires hydrogen. A bacterium capable of both aerobic respiration and fermentation, an alpha proteobacterium. When oxygen is scarce, the bacterium ferments organic matter, produces hydrogen as waste. The archaeon needs hydrogen. The bacterium produces it. They live in close association. The archaeon benefits from bacterial waste. The bacterium benefits from the archaeon consuming the hydrogen, removing a toxic byproduct. Physical proximity becomes advantageous. Over time, the archaeon's membrane protrusions might wrap around the bacterium, keeping it close. Eventually, full enclosure. The bacterium is inside. But recent research suggests the initial interaction may not have been cooperative. More likely, parasitism or predation. One organism exploiting another. Endosymbiosis often evolve from exploitative relationships, not from mutualism. The cooperation came later. So now you have a bacterium living inside an archaeon. Two organisms, two genomes. This is unstable. The bacterium has about 2,000 genes, many redundant with the host. Maintaining two copies of the same gene is wasteful. Selection favors deletion. Over millions of years, bacterial genes transfer to the host cell's genome. Lateral gene transfer. The host nucleus accumulates bacterial DNA. Modern mitochondria have only 13 to 37 genes remaining. The rest, over 1,500 genes, have moved to the nuclear genome. Why not transfer all genes to the nucleus? Hydrophobicity. Some mitochondrial proteins are so hydrophobic they can't be transported across membranes after synthesis. They must be made on-site. Mitochondria are now dependent. They cannot live independently. Their genome is too small. The nucleus encodes mitochondrial proteins, but mitochondria must import them. Complex protein import systems evolved. TOM complexes, translocase of outer membrane. TIM complexes, translocase of inner membrane. Mitochondria need the nucleus for most of their proteins. The nucleus needs mitochondria for energy. Neither can survive without the other. The merger is complete.
Here's what the mitochondria provided. Energy localization. Instead of generating ATP only at the plasma membrane, a eukaryotic cell has hundreds or thousands of mitochondria, each one an independent power plant. A human cell might have 2,000 mitochondria, each with its own electron transport chains. Suddenly, the cell has access to vastly more ATP generating capacity, not limited by surface area. Prokaryote. Approximately 50 ATP synthases per gene. Eukaryote. Approximately 5,000 ATP synthases per gene. With that energy, complexity becomes possible. A nucleus. Expensive to build and maintain. Endoplasmic reticulum. Requires energy to synthesize proteins and fold them. Golgi apparatus.
protein modification and trafficking. Cytoskeleton, dynamic assembly and disassembly of actin and tubulin. Only cells that possessed mitochondria had the bioenergetic means to attain eukaryotic complexity. You can't build complexity without the energy to support it. And you can't get that energy without mitochondria. But you can't acquire mitochondria without already having some complexity, like membrane flexibility. The Asgard archaeon had just enough complexity, membrane protrusions, cytoskeletal proteins, to enclose the bacterium. And once enclosed, the energy boost allowed runaway complexification.
Every eukaryote descends from that single merger. Eukaryotes are monophyletic, one origin, one ancestor. Every tree, every animal, every fungus all share the same mitochondrial ancestor, mitochondrial DNA, all descended from alpha proteobacteria, the genetic code. Slight variations in mitochondria matching bacterial variants. The double membrane. Inner membrane bacterial, outer membrane from host. Some eukaryotes modified their mitochondria. Hydrogenosomes in some anaerobic protists. Mitosomes in parasites like Giardia. Reduced, simplified, but traceable back to that original bacterial endosymbiont. About 1.5 billion years ago, some eukaryotes did it again. A eukaryote engulfed a cyanobacterium. Plastids, chloroplasts, the origin of plants and algae. The host was already eukaryotic, already had mitochondria, already had the complexity to pull it off. Mitochondria first, plastids later. So let's come back to you. Every cell in your body contains this ancient partnership. Your mitochondrial DNA is only inherited from your mother, a direct matrilineal line going back 2 billion years. About 40 trillion cells in your body, each with hundreds of mitochondria, quadrillions of mitochondria, all descended from that one bacterium that was engulfed by an archaeon. Every second, your body consumes about 10 to the 26th power molecules of ATP. 100 septillion ATP molecules. Without them, you would die in seconds. You are not a single organism. You are a community, a society of two ancient lineages forced into cooperation by circumstance. And the same is true for every complex organism on Earth. The biosphere's complexity exists because of one merger, one impossible event. 2 billion years ago. Evolution doesn't innovate from scratch. It repurposes, it co-opts, it merges. The greatest leap in the history of life wasn't a mutation, it was a merger. The origin of eukaryotes is biology's hardest problem. Not because we lack data, but because the event was so singular, so transformative, that it erased its own history. No intermediates survived, because intermediates couldn't survive. You either have mitochondria and the energy for complexity, or you don't, and you remain simple. For 1.5 billion years, life was prokaryotic. Simple, small, efficient. Then one archaeon and one bacterium merged, and in doing so, created the blueprint for all complex life. Every forest, every ocean, every organism larger than a millimeter. All because of that merger. Next time you breathe, remember, the oxygen you inhale is used by mitochondria, descendants of bacteria, to generate the energy that powers your thoughts.
You are the product of an impossible merger, a two-organism hybrid, and you carry that ancient partnership in every cell of your body. This is Cosmic Canvas, where we trace life's hidden thresholds. Until next time, remember, evolution's greatest leap wasn't a mutation. It was a merger, and it echoes in every breath you take.